The following program is brought to you by Caltech.
Good evening. I'm Jackie Barton. I'm chair of the Division of Chemistry and Chemical Engineering, and I'm delighted tonight to introduce Shuo Shan, who's a professor of biochemistry in my division. Professor Shan is a biochemist who's focused on understanding the chemistry that underlies our cellular machinery. We think of proteins in our cells as the workers. Now, these proteins are synthesized on the ribosome in the cell as a linear sequence of peptides. But those peptides, that sequence of peptide, has to fold up into a functional protein. How does that folding occur? And then, once folded, that protein has to go across perhaps a greasy cellular membrane. How does that happen? What's the chemistry that underlies that? How is that protein then transported to the particular organelle, trafficked to that organelle where it carries out its function? These are all actually chemistry questions, and Professor Shan has carried out elegant biochemical and biophysical experiments to elucidate this chemistry. Professor Shan received her BA at the University of Maryland, and then her PhD at Stanford, working with Professor Hirschlag, followed by a postdoc with Peter Walter at UCSF as a Damon Runyon Foundation Fellow. She joined Caltech as an assistant professor in 2005, and I was delighted when, as chairman, I got to tell her that she was promoted to professor in 2011. Among her awards, which are many, Professor Shan was a Beckman Young Investigator, a Packard Fellow, a Dreyfus Teacher Scholar. She also received the Irving Sigel Young Investigator Award from the Protein Society and the Nobel Laureate Signature Award for Graduate Education from the American Chemical Society. I'm delighted to welcome her now, and I hope you will join me. Professor Shan.
So can you all hear me? All right. So let me be here. Maybe I'll spend a little time and tell you a little more about myself. I was born 40 years ago in Shanghai of China, and I grew up there happily until I was 18. But at that point, I decided that I want to come to the United States because I want to study biochemistry. Now you might ask, what does an 18-year-old mean by that? Okay, so it was late in high school. In our biology classes, we start to talk about Mandela genetics, where Mandela, by crossing all the pea plants and observing the patterns of inheritance in terms of the color of the peas, the color of the flowers, the size and shape of the peas, or the height of the plants, somehow he deduced that there are units of heredity which he named genes, and the distribution of these genes gives rise to the pattern that he's seeing. On the other hand, in chemistry classes, our teachers started to tell us about the structure of molecules in our cells, proteins and nucleic acids, and how DNA is two strings of nucleic acids paired together by hydrogen bonds. And if you melt those hydrogen bonds, each of those strands will make a template that can make a copy of itself and then be passed down to daughter cells. So it just suddenly dawned on me that, in fact, all of those wonderful things that we see in life, colors, patterns, shape, size, heredity, the enormous diversity, and yet the extreme fidelity of all that can be explained by drawing down chemical structures on a piece of paper and then just thinking about the principles of chemistry and biology. So that was what I thought was biochemistry. It's that wonderful bridge that connects what we know about biological molecules, their capabilities and limitations driven by the laws of nature, to all of those wonderful phenomena in life. So, of course, there's a lot more biochemistry than I knew back then. And very soon I learned that although DNA codes are genetic information, by themselves, DNA do not provide function. And in order to gain any biological function, these genetic codes in the DNA has to be transcribed into another kind of nucleic acids called the messenger RNA. And then those codes in the messenger RNA has to further be decoded by a protein synthesizing machine in our cell called the ribosomes, and that's where the language of nucleic acids is transformed into the language of a linear chain of amino acids that we call proteins. And it's from this protein synthesizing machine that I would like to start my story today. Okay, so here again is where our genetic information gets decoded and translated into proteins. And every second, tens of thousands of new proteins are made in almost every single cell around the planet. And it's easy to appreciate why making protein is very important, because they then go down to do a lot of useful things. So they build our muscles. Here are the collagen fibers that comprise our joints, nails, and hair. And the vast majority of the proteins that we make, in fact, over 95% of them are actually enzymes that carry out the myriad of biochemical transformations in our body. So these proteins are really the workhorses and the building blocks that actually allow us to survive and function. So it's easy to appreciate why making protein is important. But much less appreciated is the fact that in that first few seconds to minutes, when new protein is made on the ribosome, it's when a lot of important decisions have to be made. And just like the first 3 years of a child's life has a profound impact on his or her future, I think most of parents will agree with me. Those first few seconds to minutes when new protein is made is going to a lot of decisions are going to be made that profoundly impact the future well-being of this protein, because those decisions are going to affect how this protein is going to fold into the correct three-dimensional structures, very much like folding of an origami, and where in that cell it's going to go, and who is the mailman that takes them to that correct address. How these proteins then interact with small molecules, with other proteins, or with nucleic acids to assemble into larger machine-like structures, just like the ribosome. And last but not least, how soon can these proteins be cleared away if that process becomes necessary? So a lot of the efforts in our lab are now dedicated to understanding how these decisions are made efficiently and accurately at those precious first few moments of a protein's life, which then ensures the proper balance of all the cellular proteins. So today, what I want to do is to tell you two recent and interrelated stories from which you can gain appreciation of how those decisions are made in the cell, and also gain appreciation, perhaps, for what the life of a protein biochemist is like. And let me start with a story about how those decisions are made to shuttle a very large fraction of our cellular protein to the correct cellular destination. So, a constant challenge for life is to fight against entropy and build an amazing degree of order and organization. And part of that order and organization derives from the compartmentalization of all the cellular contents into different subcellular organelles. So here's the nucleus that stores our genetic information, the mitochondria that mediates most of our metabolism, and these endoplasmic reticulum and the Golgi apparatus are these extensive membrane structures in the cell through which all the protein flow in and out of the cell, and where a lot of the protein become mature. The key here is that each of these compartments actually contain a unique set of proteins, which then endows upon the compartment the unique structure and functional roles. On the other hand, the vast majority of cellular proteins are initially made in the cytosol on these ribosomes. They now become little dots in this picture. So that creates the fundamental protein localization problem. After the vast majority of cellular proteins are initially made in the cytosol, they have to be efficiently and accurately delivered to the correct cellular destinations. Now, this problem is not only intellectually interesting to us as scientists, but also pragmatically interesting to most of you, because mishaps in these processes lead to devastating consequences. So here's a picture of the lung from a normal person where all of these airways are clear. Now, this is the lung of a cystic fibrosis patient. What happens is that there's a protein, the cystic fibrosis receptor, that should sit on the cell membrane, and it mediates a flow of chloride ions in and out of the cell. And patients with cystic fibrosis carry mutations in that protein that prevents them from getting to the cell surface. So these patients have a lot of problems regulating their body fluids, and the most devastating consequence is these lungs clogged up with thick layer of mucus. And to add injury to insult, that mucus becomes the breeding ground for bacteria. So most of these patients die out of secondary bacterial infections. Here's another example are these beta pancreatic cells. So these are literally factories where most of the 90% of protein they make are destined to be exported outside the cell and supply our whole body. And the most prominent example probably is the insulin protein that regulates our sugar uptake after every meal. So there are actually a lot of mutations isolated in the insulin gene that prevents them from properly get secreted outside those cells. So these patients develop diabetes at a very young age. So the ability of a cell to take the right proteins to the right place is enormously important for its survival and proper functioning. And to explain how that happens, it was 40 years ago that Bobo proposed his signal hypothesis. And what he basically said is that the cellular destination of protein are determined by a short stretch of amino acids called signal sequences. So that's where the X that marks the spot. And these signal sequences are literally amino acid zip codes that tells the cell which address they're supposed to go. And these zip codes are then recognized by dedicated protein targeting machines in the cell, which then deposit them to the right address. And despite the amazing simplicity of that idea, this turned out to be a general way that a lot of cellular proteins are delivered to the proper cellular compartment. So with that, Bobo won his Nobel Prize. And for this process that he initially envisioned, which is the delivery of proteins distinct for the membrane of a secretion to the endoplasmic reticulum, there's a molecular machine in our body called the signal recognition particle that carries out this process. And again, a lot of beautiful discovery biochemistry long before I arrived in the field has pieced together a rough picture of how it happens. It's a process that happens very early when a nascent protein just starts to emerge from that translating ribosome. And if this nascent chain is tagged with what we call that signal sequence, it gets recognized by the SRP. And then a series of molecular jigsaw puzzles have to happen. The SRP has to find the receptor, and the receptor has to find a specific channel on the membrane. And through this process, the nascent protein together with the whole ribosome are deposited onto these channels in the membrane, where they can finish their synthesis and gets either integrating to the membrane, or they cross the membrane and move on to the secretory pathway. Now, this is a really ancient machine. You can find it in the most remote bacteria. You find it in almost every animal in the zoo, and you'll find it in us. And without it, no cell gets to live. And like many other machines in the body, and it's mind-boggling in what it managed to do. The first is the enormous efficiency. So our cellular membranes are so important to us that roughly 1/3 of proteins encoded in our genome are distinct for these membranes. So the SRP and its receptor, although they are not very abundant components in the cell, they mediate a huge amount of protein flow. And all of this has to happen really quickly, because within 3 to 5 seconds, that nascent polypeptide keeps getting longer, and it will become too long to enter this pathway anymore. Now, if that is not challenging enough to you, there's the couple problem of specificity, because this SRP has to look at over 20,000 ribosomes in the cell, each of them making probably a different protein. So it's intriguing to think that this is 40 years after the signal hypothesis, and we have done a beautiful job identifying lots of protein targeting machineries and pathways like the SRP. But we still cannot completely understand how any of these machines manage to pick out just the right set of proteins for delivery. And that was a challenging problem for a number of reasons. The first is that these amino acid zip codes that we call signal sequences are extremely scrambled. So here are two proteins that both have to go to the same membrane through the SRP. But the signal sequences are extremely divergent in their length, shape, and amino acid composition. And don't worry that you can't see a pattern, because we haven't managed to after 40 years. And the only thing that we can deduce is maybe that these sequences are very rich in greasy amino acids. What this says is that these signals presented to the SRP is extremely noisy and scrambled. And the SRP, as a mailman, has to be extremely adaptable and be able to recognize that all of these scrambled codes for the same address. And to make things worse, here's a wrong cargo for the SRP, and its signal sequences doesn't look that much different from these correct ones. This is pretty crazy, right? I mean, I live in 2155 South El Molino Avenue, and little did I know that when I moved in that there was another 2155 South El Molino place a few blocks away. So our mailmen often deliver their mail to us and vice versa. But if you imagine that the SRP start to make mistakes like that, it's going to be very deleterious for the cells. So how do we avoid all of those mistakes? So this problem was shelved for decades. For decades, we just thought or assumed that maybe the SRP just does not interact with all these wrong cargos with no signal sequences. So these wrong cargos never enter this pathway. And this problem had to be shelved because for that long, we didn't really have a very good way of looking at how well the SRP interacts with all of these ribosomes. So eventually, it took us 5 years and a really talented and courageous postdoc, Ishu Saragi, to really solve this problem. And what Ishu managed to do is to use the latest technology in chemical biology to trick these ribosomes into accepting non-natural amino acids that are fluorescent. So as a result, a fluorescent probe gets incorporated into a specific site in that nascent protein as it's being made by the ribosome. So you can imagine that now we have a fluorescent cargo for the SRP with which we can study the interactions with each other using light, and we can even track the fate of that ribosome as it moves along the pathway. The other thing that we have to do then is to make the right and wrong cargos for the SRP. And when we started out, we didn't really know what they are. So my first student, Xin Zhang, just decided to make a bunch of them. So he very cleverly started with that borderline 4A cargo. And from there, he either increased the content of a very greasy amino acid called leucine to make stronger signal sequences, or he can do the opposite to make weaker signal sequences. And another way to make a wrong cargo is to take advantage of this protein identified before. Now, this guy has what seemed to us to be a pretty good signal sequence comparable to this third guy from the top. But as Harris Bernstein has shown years ago, this protein somehow escapes the SRP pathway for reasons that we couldn't understand. And of course, we always need a negative control, that is a cytosolic protein that should never engage the SRP. So we're finally ready to answer this question. How well does the SRP interact with all of these ribosomes? And the answer is very unexpected. So what I'm showing here are the physical binding constants, and that describes how well the SRP interacts with all of these different ribosomes with different signal sequences. And what these constants mean, literally, is how much SRP do you need to bind up half of those ribosomes. So the weaker the interaction, the more you need, and the larger those numbers tend to go. And what you see here is that indeed the strongest cargos can interact very well with the SRP, and that interaction gets 100 times weaker as the signal sequence gets weaker. But there's a very big catch here, and that is even with the weakest cargos or with the empty ribosomes, we still see way too good of an interaction. Remember, these binding constant means how much SRP do you need to bind half of those ribosomes. In the cell, you have at least 5 times more than what would be needed. And so inside the quality environment of the cell, there's a lot of chances for those wrong things to get loaded onto the SRP. And here's our biggest surprise. Is this ESPP that we know is not the right cargo for the SRP, but it's binding it very well. So if these binding constants are not very intuitive to you, here's another way to look at it, is that we can take those quantitative binding constants and the cellular concentration of all of those components and just mathematically calculate what fraction of each of those cargos are bound by the SRP after this initial interaction. And as I just told you before, more than half of the weakest cargos, in fact, over 70% of them would be bound by the SRP after this initial interaction. So what this literally said is that, in fact, the SRP makes a lot of mistakes when it first takes a look at these ribosomes and the signal sequence. And we have to propose then that these mistakes have to be corrected somehow by the cell. The question is how? And that question led us on a very long journey to figure out how the SRP then go on to interact with its receptor on the membrane and how it does so using a special protein module called the GDPases and the cellular form of energy. So let me explain that a little bit. So the most common form of energy that our cell use come in the flavor of these small molecules, guanosine triphosphates or GTP. The other type you will see is adenosine triphosphates or ATP. And the key of these molecules is that there are 3 phosphates, and between the second and third phosphates is a very high energy chemical bond. So if you clip that bond, for example, through a hydrolysis reaction, you release a lot of energy. And both the SRP and this SRP receptor belong to this huge superfamily of proteins called GDPases because they can bind GTP and hydrolyze that bond, and in that process, harness that chemical energy to do work or to provide regulation. And this is an incredibly useful module for the cell that allows it to build all kinds of molecular devices like springs, motors, clocks, or timers. Okay. Now, when we started to study these 2 GDPases, it was actually an extremely confusing process. And I'll tell you why. So these proteins, of course, can bind GTP, and in that form, if you sit them together long enough, they will eventually form a stable interaction with each other. But by themselves, that just takes hours. This makes no sense, right? Because in the cell, the SRP has to finish this whole cycle within 3 to 5 seconds. And once this stable interaction is formed, somehow additional structural changes happens on these proteins that then activates the hydrolysis of GTP. And once that happens, this complex falls apart. So this stable GTPase interaction that takes forever to happen also has less than a second of a lifetime. So this is not a very useful cycle for biology, right? And we stared at this cycle for a very long time until we realized that the cycle has to be built this way because it's meant to be regulated. And the first breakthrough finding is that this formation of this stable interaction between the SRP and the SRP receptor is strongly stimulated by a good cargo. So what used to take an hour to happen can now take place in less than a second. Okay, so here, formation of this stable GDPase complex is very much like taking this spring and bend it into this spring-loaded structure. And the mechanics of the system is built in such a way that this is not going to happen unless it receives the cues from the cargo. Okay, that not only solved the problem of speed, but also gave us important clues about the problem of specificity. Because I just told you that formation of this stable interaction, which is responsible for delivering these ribosomes to the membrane, is a lot faster in the presence of a good cargo. Now imagine what if the wrong cargo cannot do this? Then they are going to be delivered to the membrane a lot slower, right? And that's exactly what we saw. So the two strongest cargos will mediate very rapid assembly of this GTPase interaction, taking place often in less than a second. And that speed just drops dramatically as the signal sequence gets weaker. And now between the strongest and weakest cargos, we have over 1,000-fold difference in the rate at which these ribosomes can be delivered to the membrane. So now, is this enough for specificity? So we incorporate then this additional mechanism into our mathematical simulations, and you can see that things are definitely getting better. So here, the three strongest cargos are mostly retained in this pathway, whereas more than half of the cargos that are weaker than alkaline phosphatase are rejected. So things are definitely getting better, but it's still not good enough, because we know that in the cell, these two guys are not such great cargos from the SRP. So there's something else we're missing. And that something else turned out to be the ability of this cargo also to block this cycle right here, locking these GTPases into this stable interaction, but it also prevents those additional structural changes that's going to activate GTP hydrolysis. And as a result, the hydrolysis of GTP is 10 times slower in the presence of this cargo. So this is an effect that we call pausing. So you can ask, why do these molecules want to pause? If you look at what they need to do in biology, it actually makes a lot of sense, because what pausing does is to lengthen the lifetime of this complex from less than a second to almost 10 seconds. And that gives these machines just enough time to find the right channel on the membrane and to deposit this ribosome onto this channel before GTP hydrolysis drives the irreversible disassembly of these components. And you can imagine now that if the wrong cargo cannot pause, then they are going to be rejected by hydrolyzing GTP too fast, right? And supporting this hypothesis then is this absolutely inverse correlation between the speed at which GTP is hydrolyzed and how strong those signal sequences are. So literally, the hydrolysis of GTP here is used as a timer that just tells the SRP, time is up. If you haven't finished your job, just let it go. And this timer is set a lot longer for the correct cargo than for the incorrect cargos. Okay. So now if we incorporate this additional mechanism into our simulations, things really start to get a lot better. So now we are rejecting all of these, what we know as the wrong cargos. And here's just that final predictions that we have after incorporating all three of those mechanisms. And how well did we do? Actually, not too bad. So those closed circles is when Xin went back to the cells and quantitatively measured how well each of those cargos are delivered by the SRP inside the cell. And that was a great day in the lab. Even I was really surprised that our really simple-minded models, without fudging any factors, did a really great job reproducing experimental data. So what emerges from here is actually really elegant and simple mechanism with which cellular machineries like the SRP ensure a very high degree of fidelity. And maybe the analogy here is not so appropriate for mailmen, and the analogy is closer to how you are going to build, for example, an error-free security or quality control checklines. So what you need to realize is that just like mistakes in humans are inevitable, the molecules in our body work by stochastic actions, and they're going to make mistakes at a certain frequency. So instead of relying on this first security guard to be a superhuman and never make mistakes, the strategy that nature has resorted to is simply to put additional checkpoints along the pathway so that mistakes that gets made earlier get screened out at a later step. And the system will work even better if each of these security guards actually use a different mechanism to relook at that cargo over and over again. And that is just a very general lesson that we learn from the biology all the time. And the most familiar to you is probably how our genetic information are either replicated or transmitted from DNA to RNA and finally to proteins. And in each of those processes, the molecular machines that mediate them manage to achieve a much lower error rate or much higher accuracy than even the theoretical limits. Again, because these machines has learned to break down their action into multiple steps and place a checkpoint in each of those steps. And thanks to the workings of these wonderful, beautiful machines, we don't end up with cancer at age 2, and we can build that remarkable degree of order and organization that we call life. So, I've told you, this is kind of a segue point. I just told you a story about how cells make decisions to deliver proteins to the right place in the cell. What I'm going to do next is to tell you a very related story where you can really appreciate how those decisions for the protein to localize is so intimately connected to their decisions to fold or not to fold and what kind of structure they should fold into. And let me explain that a little bit. So when protein is initially made from the ribosome, it's just a long chain of amino acids, and there's no function there. And in order for these proteins to function, they have to eventually, adopt a correct three-dimensional structure. So a lot of the dancers here are probably familiar with the term, the form determines function. And exactly the same here with the proteins. It's the correct folded structure of the protein with all the correct size and shape and surface potential with the right knobs and holes that endow them the ability to carry out their wonderful functions. And this process of turning this linear chain of amino acids into a correctly folded structure is what we call the protein folding problem. And it's very analogous to how you build up a Japanese origami game, because it's really these hundreds of thousands of interlocking pieces that all has to just come together in the right way that gives rise to these wonderful and yet diverse structures. Now, if you have played origami games, you probably also have the experience that once in a while you made a mistake and you are stuck because you piece together two wrong components and you're stuck with dead-end products. And that happens in protein folding as well. This is what we call misfolding of the protein, or even worse, this protein happens to stick with each other and form massive aggregates. Now, why are we interested in these mistakes in protein folding? Because it does happen in our cells, and when it does, it leads to devastating consequences. So here's a beautiful brown eye of a young person, and that's the eye of a cataract patient. And what happens is that that crystalline protein that make up our clear eye lenses after many years of use has refused to stay soluble, and it has crashed out and aggregated. So at this stage, the only cure is to surgically remove that lens. Now, here's a slice of the hippocampus from an Alzheimer's patient, where instead of very clear slice, what you see are these large protein deposits or aggregates formed by a protein fragment, the amyloid beta. And although the field is still debating which form of these aggregates is actually causal to the disease, most of us has agreed that the process in which this amyloid beta protein aggregates to form these large structures is highly toxic to the cells and killed a lot of our neurons that's intimately related to onset of these diseases. And just numerous examples like that has emerged in the last decades or so, where a lot of the neurodegenerative diseases, Alzheimer's, Parkinson's, Lewy body syndrome, dementia, are all related or even rooted in these mishaps in protein folding. So you might ask, how does cell avoid these devastating consequences? And the answer is that they have a special set of proteins called the molecular chaperones. The term is probably familiar to you. So chaperones refer to these old, wise, very well-behaved people who will oversee the interaction between young people and make sure that wrong interactions do not happen. And just like human chaperones, molecular chaperones in our body prevent the wrong interactions between proteins from happening, and by doing so, they guide the protein along the proper folding pathways. Now, if these devastating protein aggregates have formed, there are also a special class of chaperones that might even be able to reverse this process. So these are rare but highly useful proteins, and I'll come back to them a little bit later. So now if you think about chaperones in our body or cells, and all the cellular proteins, they are client proteins in the cell that they have to take care of. Who would you imagine are the most difficult clients? It's those proteins that are distinct for the cellular membrane. Okay, here's an example. So this is the most abundant protein on the planet. That is the protein that comprises the light harvesting complex in green plants. So these are protein complexes that sits on the thylakoid membrane of green plants, and each of those complexes will then bind up over 60 photosynthetic pigments so that the energy from sunlight can get absorbed and then transmitted through the photosynthetic centers. So this is the beautiful molecule that feeds our planet. But now if you strip away all of these colorful chlorophylls and these membrane bilayer and just look at the protein by its bare bones, you realize that there's a lot of trouble with this protein. So this is a typical way that we look at our protein, is to look at its electrostatic surface properties. And here the colors are different charges, and it doesn't even matter because this protein has very little of them. And most of this protein is made up of highly greasy amino acids. That's why it's colored in white. And grease likes to interact with grease. So this protein is meant to sit in the membrane and interact with all of those photosynthetic pigments, but as soon as it sees a water-like aqueous environment, all it wants to do is to crash out and aggregate. And in fact, that's all we saw in the first few months that we're trying to work with it. So that is just a really general problem for the cell. Again, 1/3 of our genome encodes membrane proteins. And these have highly greasy proteins. Before they arrive at that final membrane destination, they have to cross at least one and sometimes two water-like aqueous environments, and where they're highly prone to aggregation and misfolding. So how does the cell solve this problem? So here's my second graduate student, Piera Sazu-Empopon, and she has taken on this project of figuring out how this very difficult greasy protein managed to find its destination on the thylakoid membrane without mishaps. And during this process, she accidentally discovered that there's a very special component in the chlorophyll platform of the SRP that offers a very potent chaperone for this light harvesting complex. I know the names are really nondescript, so from now on, I'm going to call this thing our little chaperone. Okay, so this is probably the only raw data that you're going to look at, but the principle is pretty simple. When proteins form large aggregated structures, they scatter enormous amount of light, and you can easily read out that light scattering on any photometric device. So this protein is really so bad. As soon as you put it in an aqueous environment, it aggregates spectacularly. But if you put it into a solution that contains our little chaperone, then it completely suppresses those aggregation. And it's one of those rare experiments where I don't even need an instrument to tell me what happens. And with a lot more hard work, Piera has further shown that out of all the proteins in the chloroplast of green plants, this chaperone is both necessary and sufficient to protect the light harvesting complex and prevent it from aggregation. And if you kill that activity, these complexes do not get to the thylakoid membrane. So at this point, we got a little bolder. We asked, what if we just let this protein aggregate first and throw back this chaperone? Will it be able to reverse this process? Now, this kind of activity is very, very rarely seen, so I wasn't very hopeful. But Piera was bolder, and she said it never hurts to try. And she was right. Within a few minutes, this aggregate was completely cleared. So what's going on here? So it's time to think about models and mechanisms. On the one hand, we can imagine a very boring model where once in a while, this protein is going to fall off this aggregate. And when it does so, it gets bound and trapped up by this chaperone and cannot go back to these aggregates. And here's where mathematical simulations help. Because when you put this model into simulations, you realize that there's a very strong prediction from this model, and that is the speed with which this aggregate is cleared is reasonably independent of how much chaperone we are throwing in. And the reason is pretty simple. The slowest step in this whole process is the spontaneous falling off of this protein from the aggregate. So no matter how much chaperone we are throwing in, we're not going to affect how fast it can be cleared. On the other hand, there's a much more interesting mechanism we can imagine, where this chaperone can recognize this aggregate, remodel it, and finally disassemble it into soluble components. And of course, we need to complete that cycle. But if you put this model into the simulations, there's a totally different prediction. Because this is an active mechanism, now the speed at which this aggregate can be cleared is highly dependent on the concentration of the chaperones. So here are the predictions, and that's the data. You'll probably agree with me that the data looks very much like this chaperone is acting actively. And we got something even better than that. And if you plot how fast this aggregate is cleared as a function of how much chaperone we're throwing in, you get these nonlinear dependencies that we call cooperativity. And that might sound hogwash to you, but what cooperativity means to us is that these chaperones actually collaborate with each other. So binding of the first guy actually causes structural changes in the aggregate that allows the second and third molecules to bind even better. So together, the data is now very strong that this little chaperone can actively recognize, remodel, and disassemble a very stable aggregate formed by its client protein. Okay, now this is very unexpected. And the one reason that it's unexpected is that very few chaperones have been shown to do this, and here are actually all of those. And the names doesn't even matter. The second thing that was very unexpected is how different our little chaperone is compared to all of these previously studied systems. So if you think about the time it takes for these chaperones to accept their action, our little chaperone is not too far off from all of those other systems. But all of these other systems are just massive molecular assemblies. And they're so complex that they work by generating molecular forces using the fuel from ATP. So every time they try to pull a protein from the aggregate, they burn up hundreds of ATP molecules. On the other hand, our little chaperone is less than 1/10 of a size. And Pierre swears that she never put ATP into the reaction. And so what happens here? So after a lot of thinking and observations, this is the way that we are trying to explain it right now. So the difference fundamentally lies in the different client proteins that these different chaperones are dealing with. Well, if you look back at evolution, what you realize is that our little chaperone sort of co-evolved with the emergence of light harvesting complexes in green plants. So this is a chaperone that's dedicated to serving this most abundant family of membrane proteins on Earth. And because of that dedication, it has plenty of time during evolution to build extensive interactions with its client protein. So with that specificity comes proficiency. And in the absence of any external energy input from ATP, the energy derived from those interactions are probably sufficient to disassemble those large protein aggregates. On the other hand, all of these other chaperones has to deal with all kinds of client proteins in the cell whenever stress comes in. So they have to deal with client proteins with vastly different size and shape and amino acid compositions. And so they really have never had the chance to build extensive interactions with their client proteins. And without that, what they resorted to is just getting bigger and bulkier and use brutal forces by burning up ATP to do their job. So at the most fundamental conceptual level, what this chaperone tells us is a new concept, that maybe we have overthought how hard it would be to disassemble protein aggregates. Because, if you can dedicate the chaperone to a specific protein and build up clever enough binding interactions, those interactions should be sufficient to disassemble large protein aggregates. And this chaperone really have a lot of those binding surfaces. So this is a structural model of this chaperone built together from at least the works of at least 3 or 4 different labs. And the only thing I want to say today is this, that this is a very long molecule. So despite its very small size, it is able to offer extensive surface area to interact with its climb proteins. In fact, we have spent the next 2 years after that initial discovery to map out how those extensive surfaces are used to interact with different parts of the climb protein and how each of those different interactions are used to drive the different recognition and disassembly steps as this chaperone fights its way through the aggregate. So, this story is very typical of how science often goes. As soon as you answer one question, it raises even more fascinating questions. How does this chaperone really work? Will we be able to understand it at the level that we can accurately predict what will happen if we modify it? Given that this is a much simpler system than all of those previously studied chaperones, we're confident that this might happen. How many chaperones in our cell actually behave like this? Have we ignored them because they're small and they don't do fancy things like burning up ATP? And this is the most tantalizing thought. If there's a day that we can really understand how this chaperone works, what will it teach us about how to overcome all those protein aggregation problems that are associated with disease? Okay, and this is one of those motivating experiments that keeps us going. So here again is that amyloid beta protein that even in a test tube, it likes to assemble into massive fibril-like aggregates and huge deposits that eventually gives rise to the Alzheimer's disease. But if you mix this amyloid beta with our little chaperone, even though they come from totally different species and have never seen each other, that fibril formation and aggregation is prevented for as long as we can imagine. Now, we are not the only lab who has seen this. Many labs around the world has demonstrated the similar capabilities with the chaperones they were studying. So as a field, we know that we're very far away from the cure, but we also know that somewhere in those chaperones is the possibility or the hope of overcoming those devastating problems associated with protein misfolding diseases. If only we can understand really how those chaperones work and how to unleash their powers. So this is 20 years after I initially decided to study biochemistry. And the longer that I go along the pathway, the more I'm convinced that this is the right way to understand and at least demystify the phenomena of life. I hope I have convinced you that by understanding the property of molecules in our cell and understanding what they can and cannot do, we can start to demystify how cells build that remarkable order and organization that's essential for life, and also we can gain an understanding of how when these molecules make mistakes, they lead to devastating diseases and how we might be able to intervene in them. And with that, I will just thank all the people who did the work. So I've talked to you about the work of Ishu Saragi, Xing Zhang, and Pierre Sarou Empopon. And of course, there's a lot of work from other members of the lab that I will be happy to talk to you anytime. And what's important to recognize is that, it's often the PIs or the professors that gets all the recognition, but really it's these students and postdocs. There's sweat and tears and blood stains on the bench that gives rise to these wonderful science that we see. And so without them, we really don't have that driving force. So another wonderful thing about doing science is not only that the lab is never a boring place, but also that you get to talk to other people who are also very talented and interesting and tell you all kinds of new things that allows you to learn even more. And very often, if we have a common interest, interesting collaborations happen. So a lot of our work is deeply motivated and complemented by collaborating with scientists around the world, structural biologists, chemical biologists, physical chemists, and experts in the chaperone field that holds our hand as we march into this new field. And finally, of course, without all of this work, it would not be possible without these funding. Without the funding from these foundations. And in an age where the funding from NIH keeps getting cut and we run on a tighter and tighter budget, I really would like to thank my institution and my division, especially chairs like Jackie, who always give us relentless and unconditional support for our work. So with that, I thank you all for being here, and I'd be happy to answer questions.